embryo sac development, and another structure of embryo sac, okay, and development of tetrasporic embryo sac, different types of embryo sac in angiosperms, that is Araxa tetrasporic type and Peperomia tetrasporic type. Next, Pinia and Camaro tetrasporic type, tetrasporic type, and tetrasporic type. All these are types of embryo types of
Sir,
embryo sac.
hello, sir.
Okay. Ah,
Sir, your voice is breaking, sir.
okay. Hello.
Yes, sir.
Now okay, student. Now okay.
Yes, sir.
Okay. Okay. Yesterday, the study embryos, development, structure of embryos, different types of embryo sacs. All these are studied yesterday. Okay. Structure of embryo sac. Generally,
Yes, sir.
embryo sac shows three parts. That is, its apparatus, antipodal cell, and central cells. Okay. Upside, micropolar region, and opposite side, that is antipodal region. Antipodal side, three cells, and micropilar end shows three cells. In three, one is egg cell. Okay, egg cell shows bigger than remaining two cells. Okay, centrally located two cells. Two cells are fused. That's why they are called secondary nuclei. This is the structure of embryo sac. Structure of embryo sac yesterday studied. Okay. Hello, students.
Yes, sir.
Hello. Audible?
Yes, sir. Yes, sir.
Okay. 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 Today topic pollination unit third, number 16 to 23rd. Pollination. Okay. Pollination
Okay, sir.
means process, the process transfer of pollen grain from the anther to the stigma. Okay. So transfer of pollen. Process of transfer of pollen from to is called pollination. Okay, that is pollination. Transfer of pollen from anther to stigma is termed. Okay, this transfer help of pollinating agents. Different of pollinating is pollination, sorry, transfer agents and artificial. First, pollination in angiosperms classified into two types. That is pollination. Okay, pollination and cross pollination. Self pollination and cross pollination. Self pollination means transfer of pollen grains, transfer of pollen grains and to stalk of the same flower. It is called pollination or autogamy. Self pollination or autogamy. Answer is pollination means transfer of pollen grain from anther to stigma of the same flower is called self-pollination or autogamy. Autogamy. This type of pollination is possible only bisexual flowers. Okay, only bisexual flowers. means male and female organs located same flower. Flowers found in both clistogamous, clistogamous flowers. Clistogamous flowers means the flowers which do not open are called clistogamous flowers. Okay, clistogamous flowers. Okay, which do not open are called clistogamous flower. And chasmogamous also there. Chasmogamous flowers. The flowers which open are called chasmogamous. Do not open clistogamous. Opened flowers is called chasmogamous flowers. And the pollination is called chasmogamy. Okay. Autogamy means self-pollination. Next, cross-pollination. Cross-pollination means stop pollination from the anther of one flower to the stigma of another flower. Okay, one flower to the stigma of another flower. Another flower of same species is called cross pollination. Okay, is called cross pollination. Cross pollination means transfer of pollen grains from the another one flower to the stigma of another flower. Species is called cross pollination or allogamy. Okay, or allogamy. Okay, next the cross pollination is two types. That is gonadogamy and genogamy. Okay, gonadogamy and genogamy. Okay, gonadogamy and gonadogamy means it is type of cross pollination. A type of cross pollination. It occurs between two flowers present on the same plant. That means pollen grain of one flower fall on the stigma of another flower present on the same plant. That is gynetogamy. Next, xenogamy. If the pollen grains of the if the pollen grains of one flower reach to stigma of another flower of the same type of present on the plant, that is genogamy. Next, okay. Next, pollinating agents already that is wind insects and birds. Next, pollinating agent wind, water, insects, and bird. Okay. Next, first pollinating agents. Okay. Pollinating agents carry pollen grains from another to from anther to stigma of same or different plants. That is the function. Pollinating agent function is carry of pollen grains from the anther to stigma. Okay, carry of pollen grains from anther or stigma. And that okay. Anther means pollen grain. Pollen grain sheath. It is anther. Anther contain number of pollen grains covered by one sheath or layer. That is called anther. Okay. But pollinating is carrying complete anther. Okay. Next, two types. Pollinating agents commonly two types, that is biotic agents and abiotic agents. Okay. First, biotic agents means living organisms which act as agents of pollination. Example, insects, birds, birds, insects. Types of birds fall on flowers, then catching the feather, pollinating pollen grains, then same bird going and falling the next flower or next same or another tree or another flower tree. Then these uh, pollen grains are transferred, gradually transferred and settled down on the another plant flower stigma by birds and insects. Next, abiotic agents. Abiotic agents means non-living objects which act as pollinating agents. They wind by the help of wind and water also participated in transfer of pollen grains. Okay. Next, pollination by wind. Okay. Pollen grains are carried by wind from anther to stigma. Okay. Wind pollination is very common seen in grasses, monocots grasses. Okay. Very light weighted pollen grains are moving on wind. Okay. The characteristics of plant pollinated by wind are light pollen grains, non-sticky pollen grains, well exposed stamens, large feathery stigma. Okay, these are characters of light or wind pollination, participated in wind pollination. Next, pollination by water. Okay, this is a less common mode of pollination. Water pollination can occur in number of ways. Okay, that is water act as medium for the gamete transfer in lower plant. Example, Bryophyta, Vallisneria, etc. Okay. Next, female flower reach the water surface and stop some different type of flowers and reach the water surface by stacks where male gametes were already released. Example, Vallisneria, already told. The water pollination pollen grains are prevented from being wet by a mucilaginous covering. Next, insect. Okay, insect. Bees, for example, bees, most common biotic pollinating agents. Bees, okay, bees transferring or carrying different types of pollen grains from one flower to another flower of same species. Bees, it is common insect pollinating agent. Okay, bees. Yucca also. Okay, yucca mouth relation is mutual ones. Okay. Next, pollen pistil interaction. See here, diagrams here. Bees touching or absorbing honey, one flower. Then at the time of catching the honey, then pollen grains are settled on feathers, bees feathers and mouth and legs. Then same time and moving to surrounding flowers and same the absorbing the honey. That time transferring the pollen grains. See here the diagram touching one flower and at the same time touching remaining three flowers. Okay. Next, pollen pistil interaction. Okay. At the time of pollen grains reaching by biotic and non-biotic pollinating agents like birds, water, wind. Okay, finally reach the stigma. Okay, pollinating agents or pollen grains are reached by different methods. Okay, biotic, non-biotic methods. Finally reach the stigma of same flower or another plant flower, different species flower, finally reached. Okay, that is pollen pistil interaction. Describe the pollen pistil interaction. Pollen and pistil interaction between action. Pollen and pistil. Okay, pollen landing on the stigma of pistil. Compatible or non-compatible. Okay. After reaching the pollen on stigma, of the compatible, some of the not compatible. Okay, with the stigma action. Okay, stigma action. Next, next, pollen pistil interaction. Second, the pollen pistil. Okay, therefore, acceptance or rejection of the pollen by stigma depending on the compatibility. Sometimes some pollen grains are accepting. Okay, some pollen grains are rejecting depending on the compatibility of stigma. If the pollen and pistils are compatible with each other, okay, following events will take place. That is, pistil accepts pollen and formation of pollen tube. After reaching the pistil, formation of pollen tube. After reaching the pollen, then different types of reactions started. That is, pistil accepts pollen, then formation of pollen tube on pistil, and pollen tube reaches gradually ovary. Gradually ovary. Pollen grains move into pollen tube. Okay, pollen grains move into pollen tube. Pollen tube finally enter into ovule. Okay, ovule through micropyle. That is the main way of embryo. Okay, the main way of embryo or ovule. A pollen tube enter as pollen tube finally enter into ovule through micropyle.
Are you okay, sir?
Next, pollen tube finally pollen tube enter into micropyle. So pollen tube enter through filiform apparatus. Then finally fertilization. Okay. If pistil rejection pollens if non-compatible. Okay. Okay, if compatible, then pollen tube started and moving towards ovule, then finally reach the ovule, then fertilization. If, on the other hand, if the pollen and pistil are incompatible with each other, means stigma and pollen, the following events will take place. That is, Pistil rejection. Pistil rejects pollen. Prevent pollen tube growth. Okay. If not reject, pollen tube growth started. If rejected, pollen tube growth prevented. Okay. And same time, prevention of fertilization. Okay. Fertilization also prevented. The pollen crystal interaction can be defined as follows. That is, pollen crystal interaction is a dynamic process of pollen recognition followed by promotion or inhibition of the pollen. The chemical components of the pollen interact with those of pistil. Okay, that is different types of chemical shows. Secondary zone. Okay, secondary zone. Okay, pollen shows different um, places or different parts. That is central cavity and stigma, exudate, papillae, secretory zone, storage zone, vesicular trace, stigmoid tissue. All these are the stigma parts, surface of the stigma parts. Okay, this is the stigma development. Next, pollen tube after receiving the stigma, then start pollen tube growth. Okay, pollen tube growth. Okay, pollen tube growth. Pollen showing different material. Okay, means cytoplasmic particles and chemical constituents. That is lipid. Pollen tube having a lipids, starch, protein, and mitochondria, polysaccharides, RNA. Okay, polysaccharides and RNA. All these are having pollen tube. Okay, especially lilium showing different cytoplasmic particles. Means lipids, starch, protein, mitochondria, polysaccharides, RNA. Okay, all these are pollen tube materials. Pollen tube materials. Next, next, at the time of moving, at the time of moving pollen tube from stigma to ovule. In that place, pollen pistil or style is commonly shows closed style and hollow type of style. Style two types: closed style and hollow type of style. Okay. Generally, female organ shows pollen grain or stigma, style, ovule. After receiving the pollination, means carrying the or receiving the pollen grain from stigma to style, then pollen grain gradually moving towards ovule. At the time of reaching the ovule, pollen tube reached three ways. That is porogamy, calagogamy, and mesogamy. This is pollen tube growth. Okay, see here, this is micropyle region. This is integuments. Centrally, showed pink color part is embryo sac. This is ovule. Okay, pollen tube coming from stigma, then enter into style, then style to gradually moving to ovule side, ovule region in three ways. First, porogamy. Porogamy means the entry of pollen tube into ovule through micropyle. This is porogamy. See, pollen tube enter into ovule through micropyle. This is micropyle region. See diagram here. This is porogamy. Okay, remaining two types also there. That is collagogamy. Okay, in some plants, the pollen tube enter into ovule through collagen. Collagen means upside. Okay, antipodal cells is there now. Embryo sac upside, that is collagen. Okay, pollen tube enter into collagen, that is collagogamy. Next, mesogamy. Okay, pollen tube in some plants, sometimes pollen tube enter into the ovule through the integuments. Means pollen tube enter into the side by side. That is mesogamy. That is mesogamy. Okay. Finally, pollen tube enter into ovule. Then fertilization process is over. At the time of entry of pollen grain or pollen tube enter into the ovule, then fertilization is over. Okay, that is syngamy. Syngamy means pollen tube having two male gametes. Okay, two male gametes. Okay, one gamete or one spore, male spore. Okay, after entering of ovule, then embryo sac is there, ovule. Then one male gamete enter and fused with egg. Fused with egg. That is called syngamy or fertilization. Okay, pollen tube enter into the ovule. Then tube ends completely burst or degenerated by apical pores. Okay, by the formation of apical pore. Then released two male gametes. Pollen tube having two male gametes. One is then tube bursted in ovule. Then one male gamete going and fused with egg. That is fertilization or syngamy, discovered by second male gamete going with secondary nucleus or polar nucleus. Polar nucleus, two nucleus are fused now. Then remaining another nucleus or male gamete also going and fused with two polar nuclei. Okay, two polar nuclei. Then this results the formation of triploid. Formation of triploid. Okay, triploid means primary endosperm nuclei. Pen is formed. Okay, this was discovered by Navashin in Lilium and Fertilaria plants. Lilium and Fertilaria plants. Triple fusion, commonly called triple fusion. Okay, first male gamete going and fused with egg, that is fertilization or syngamy. And second male gamete after bursting the pollen tube and moving towards and fused with secondary nuclei. Okay, secondary nuclei, where is located? In embryo sac, central portion of the embryo sac. Already two nuclei are fused, that is secondary nucleus. And another third nuclei also going and fused. Then here three nucleus is there. That's why called triploid or triple fusion. Okay, commonly called triple fusion. Here commonly called triple fusion. Okay, first sperm nuclei combines with the egg cell form, and other the secondary nucleus to form primary endosperm nucleus. Okay, with this occurrence of two fertilizations. Okay, first one is egg, second one is uh, secondary nucleus. Okay, that is first fertilization. Secondary nucleus fusing is second fertilization. Okay, two fertilizations is occurred. Okay, the two fertilization in this phenomenon is described as double fertilization in embryo sac. Double fertilization. Double fertilization includes singamy as well as triple fusion. Singamy as well as triple fusion. It results in the formation of fertile endospermic seeds. Okay, fertile endospermic seeds. Okay, students. First one is in embryo sac showing two fertilization. Okay, after entry into the pollen tube, entry into the embryo or ovule, then pollen tube bursted in ovule. After bursting, releasing two male gametes. Pollen tube having two male gametes. After bursting in ovule, releasing two male gametes. One is fused with egg. Second one is fused with secondary nuclei. Okay, secondary nuclei having already two nucleus are fused. Then another one is joined. That's why triple fusion. Triple fusion. Okay, one is fused with egg cell. That is first fertilization, and second fertilization means second gamete going and fused with secondary nuclei. That is second fertilization. Completely, but two fertilization is over here. That's why they are called double fertilization. Double fertilization. Okay. In first fertilization, form a zygote. Okay. Second fertilization, form a endosperm. Endosperm. Okay, students. So this is no need for slides. Endosperm. Okay, next endosperm. After in angiosperms, after fertilization, different types of changes occur in the flower. Okay, these are called post-fertilization changes. Post-fertilization changes. The sepals, petals, stamens, style, and stigma fall or fall off. But in some members of Solanaceae family, and the calyx remain persistent. Okay, even after the fertilization end, grow along with the fruit. Persistent means not removed. Okay, after post fertilization changes. What is the post fertilization changes in ovule pods? For example, funicle changes after fertilization. Funicle changed into or converted into stalk of the seed. Next, outer integument, testa, seed coat. Inner integument, stigma, inner seed coat. Micropyle change converted into after fertilization. Then zygote after fertilization converted into embryo. Next, synergids degenerated. Okay, upside two synergids is there now. Upside. Okay, embryos upside. Synergids is there. The type of synergids completely degenerated. Okay, completely degenerated. Next, primary endosperm nuclei. Primary endosperm nuclei. Pen. Pen is converted into after fertilization endosperm. After fertilization endosperm. Okay. Xylem means hair on the seed. That is xylem. All these are after or post fertilization changes. Post fertilization changes. Okay. Next, endosperm. Okay. Next, endosperm. Development of embryo and embryogeny. Next, endosperm. Okay. Endosperm. Okay, this is unit 4 endosperm and endosperm types. Unit 4 endosperm, block 3, unit 4 endosperm, page number 27 to 32 endosperm. Okay, endosperm means after fertilization. Okay, double fusion is over. Normally, angiosperms, the endosperm is formed from primary endosperm nucleus after fertilization. Okay, secondary nucleus. Okay, pollen tubes having two nucleus is there now. Two nucleus released in ovule. First nuclei going to egg and fused second nuclei and fused with secondary nuclei. Secondary nuclei is triple fusion. Already two nucleus is there. Then third nucleus also come and join. That's why we are called triple fusion. Okay, triple fusion after some time form a endosperm. Okay, after some time form a endosperm. Okay, endosperm is the nutritive tissue. Endosperm is a nutritive tissue. Okay, this is endosperm. Endosperm is a nutritive tissue. Okay, useful for the developing embryos. Useful for the developing embryos. Okay, it is already triplet condition. We know, already discussed 2-3 times. Endosperm is triploid condition. Triploid condition means pollen tube having two male gametes. Okay, released in ovule. Okay, after releasing one male gamete going to already secondary nucleus is there in embryo sac. Okay, secondary nucleus means two nucleus are fused. Then another extra nuclei is or male gamete is also go and fused. That's why triple condition or triploid 3N condition. That's why called 3N condition. Endosperm is 3N condition. Triploid nuclei form a endosperm. Endosperm is nutritive tissue. This is always showed triple N condition. Triple N condition. But in gymnosperms, the endosperm is developed directly from female gametophyte. In gymnosperms developed directly from female gametophyte before fertilization. Hence, it remains in applied condition in gymnosperms only. Okay, in gymnosperms only. Okay, in some plants during the developmental stage, the embryo takes food from the endosperm. Okay, endosperm is a facilitator. Okay, food facilitator for embryo. Okay, embryo and completely utilized. Okay, completely food utilized it by the time on the ovule is converted into seed. Okay, endosperm completely utilized and then converted into seed. Okay, at that time ovule contains only embryo. Endosperm is empty, means embryo completely utilized like food. Then endosperm is completely empty. That time only remain part is embryo. Okay. The without endosperm, the type of seed is called non-endospermic seeds or exalbuminous seeds. Exalbuminous seed. For example, Dolichos Caesar capsula. Okay. Dolichos Caesar capsula. Next, in some plants, embryos cannot utilize food like a food. Endosperm cannot utilize like a food. Okay. Some amount of endosperm is there. The type of seed is called endospermic seeds. Not utilized is not. Endosperm is not. Endosperm, embryo sac not utilized endosperm like a food. That's why endosperm is there. The type of seeds is called albuminous seeds or endospermic seeds. Okay, albuminous seeds are endospermic seeds. Okay, next formation. Next, the formation of uh, uh, seed. Formation of seed and embryo. Okay, first embryo formation. Okay, embryo formation means different phases or different types is there. Okay, different types is there. That's why we are studying one by one. Endosperm studies one by one and types. First types. Okay, depending. Okay, depending upon the mode of development, the three types of endosperms recognized have been recognized. That is, first one is A, nuclear endosperm, B, cellular endosperm, C, helobial endosperm. Nuclear, cellular, helobial endosperm. Okay. Cellular endosperms is largely restricted to dicotyledons families. Okay. Helobial endosperms, monocotyledons. Okay. This is the okay. Oospores, endosperms development here. First, two-celled embryos first. Okay. The zygote okay here types. Okay. We have studied endosperm typing. Okay. First nuclear endosperm. Okay. The nuclear endosperm development is different in different taxa. Okay, in some plants, for example, coffee walls appear at the 4-nucleate stage. Okay, 4-nucleate stage. The wall formation in many free nuclear taxa is delayed until many nuclei are formed. Here, one to many nuclei is formed. Okay, upper side embryo. Okay, Next, downside, embryo endosperm, centrally hollow place. Finally, nuclear embryo endosperm is living. Okay, the free nucleus enveloped in cytoplasmic sheath. See the diagram. This is a nuclear envelope formed on cytoplasmic sheath. In some species, endosperm remains nuclear. The cellular phases does not appear. Okay, that is nuclear endosperm. Next, cellular. Okay, cellular endosperm. The free nuclear stage is absent in this type of endosperm development. The every division of the primary endosperm nucleus is formed by cell wall formation. Okay, formed by formation. So that the endosperm is cellular from the beginning time. Okay, the first division of primary endosperm nucleus is transferred along with the embryo sac. Also get divided into two by transfer walls. Finally, form a cellular endosperm. Next, helobial. Helobial endosperms. See here, helobial. This type of endosperm development is intermediate to cellular and nuclear type. Intermediate cellular and nuclear type. Okay. The primary endosperm nuclei divide in situ after comes to the collageal end. Okay, this is collageal end shows embryo. And near the antipodal wall formation takes place, producing large microphylar and small collageal number. The collagen number is densely cytoplasmic deep staining. Okay, this is the helobial type of embryo sac. Okay, helobial type of embryo sac. See here, two-celled embryo and endosperm nuclei. Okay, first two division, then form a complete cellulose or nucleus filled with sheath filled with nucleus. Okay, this is nuclear endosperms. Okay, this is example: embryo, cellular endosperm, and free zone. This is also okay. Next, cellular endosperm over. This is cellular endosperm. This is Ladin ramitia plants. Helobial also over. Next, embryo sac development. Embryo sac development. Okay, embryo sac development is okay. Embryo sac development. Where is embryo sac development? Okay, here embryo sac development is there. It is first development of embryo. Okay, embryo or embryoni. The syngamy means producing diploid zygote. Okay, syngamy means diploid zygote. The zygote divides in highly organized and predetermined manner. Okay, zygote start with so large vacuoles. Okay, large vacuoles. Okay, endosperm page 27 to 27 to 37. Okay, 27 to 37. That is endosperm. Okay. Okay, endosperm is three types. Again, endosperm is three types. That is erobial, nuclear, and cellular endosperms. Erobial, nuclear, and cellular endosperm. Okay, this is three types of endosperms. Once again, see here, endosperm of Stechausia showing cytoplasmic nodules. See here, cytoplasmic nodule. Okay, first embryo, then endosperm wall is started. Endosperm wall is started. Gradually, in cytoplasm filled with nodules, nucleus sheath, nuclear endosperm, nuclear endosperms. Cellular over already. The cellular of nuclear stage. Absence of three nuclear stage. Okay. Number E. Okay. The endosperm formed the mentioned earlier nucleus of the basal apparatus of hypertrophy. Last stage, this nucleus shows repeated endomitotic base. Astoria means condition for lower endosperm below portion form a thread-like structure is called endosperm hastoria. Endosperm hastoria. Okay, that is then grow. Next, three types of endosperms we discussed means next endospermic seeds and non-endospermic seeds also discussed. Non-endospermic seeds are cellular endosperm also discussed. Okay, cellular and cellular endosperms, cellular endosperms, and three types is there na nucleus, cellular. Next, hello students.
Yes, sir.
Next, Hilobial endosperm, sir.
Yes, yes, good. Next, here, endosperms. All types of endosperms form a ball. Nucleus, all are formed on the wall of the cell of endosperm. Endosperm main function is, what is the endosperm main function? Hello, students. What
Sir,
is the
your
main
voice is
function?
breaking, sir. Hello, sir. Your voice is breaking.
Nutrition. Okay. Seeds, two types of seeds, that is endospermic and non-endospermic seeds. Embryo used completely, okay, embryo completely as a food material. Embryo used as a food material, okay, completely used. Then endosperm is going to empty. The type of seed is endospermic or albuminous seeds. Embryo sac not utilized as a food. Okay. Then the type of seed shows endosperm. The type of endosperm, the type of seed is called endospermic seed. Okay. Hello. Hello, students.
Yes, sir.
Yes,
Yes,
sir.
sir.
Okay. Seeds are how many types? Endospermic seeds.
3.
Three types.
Okay, good. Seeds two types.
Cellular, nuclear,
Endospermic two types.
Hilobial.
Okay, good. Okay, next topic, embryo. Okay, next topic, embryo. Tomorrow we will discuss. Okay, okay, students. Hello.
Yes, sir. Yes, sir.
Okay, thank you.
Thank you, sir.
Thank you, sir.
Thank you, sir.
Okay, okay, bye.